Hello, everybody, and welcome to our question lab this evening. We're happy to have you here. As you guys know, we do this every Tuesday at 6 p.m. And today we will be covering genetics. So today we're covering genetics. It's a topic that you guys probably haven't studied much since you first learned it in medical school, which is why we're doing it today, because even though it may have been a while, it can still appear on your test and you need to have a good understanding of it. So with that being said, my name is Sean. I have been with COMLEX for about 1.5 years, working with RX Coach. And prior to that, I've been working with students in test prep for the better part of almost a decade. So a lot of experience when it comes to USMLE Step prep, helped a lot of students achieve their goals, and we hope to help you as well. We have some wonderful panelists joining us today. One of our panelists is one of our RX coaches. If you haven't heard about RX Coach, I'll talk about that here in a little bit. And that RX coach is future Dr. Ari Stone. So I'll let Ari introduce himself.
Hey guys, welcome back. So my name's Ari. I am, like Sean said, an RX coach. I've been for about 3 months. I'm a 4th year at the University of Wisconsin going into ENT. And it's nice to see you guys back tonight.
Excellent. In addition to that, we have Kate and Jeff joining us today. If you guys look at your control panel, there should be a little question box there, and that's where you can ask questions to our wonderful panelists, and they will do their best to try to answer all of your questions. Joining me in my discussion today is Mr. Jeff Downing. Jeff, you want to say hello?
Good evening, everybody. Yes, this is Jeff Downing. I am the Director of Product here at COMLEX. I've been in the world of educational technology and helping prepare healthcare professionals for quite a while, over 15 years. So I oversee our product development and our marketing program here at COMLEX and USMLE-Rx, and love being a part of our weekly question lab. So looking forward to this one.
Excellent, and we're happy to have everybody here. Since you guys had a chance to meet us, we'd love to get a chance to meet you guys. I know that generally speaking, a lot of you guys are studying for Step 1, so I'd like to know, when are you guys planning on taking Step 1? Are you guys taking it this fall, this winter, sometime next year in 2022, or are you just not sure? Take a second to select your answer. This is the same exact type of format that'll pop up after all of our 4 questions this evening. Well, you'll have a chance to give us your responses to what you think the correct answer is for each question that we do. So familiarize yourself with the format here if you haven't done so already, if this is your first time. I see a lot of repeat attendees in attendance, so I appreciate you guys coming back. Also see some new names, so for those of you who are new, welcome to the question lab. We hope to see you in subsequent sessions. Next week, we will be covering neuropath next week. All right, let's see here. So it looks like about 30% of you guys are taking it next year. We've got 41% of you guys taking it this summer, and some of you guys are taking it here within the next couple of months in August and September. So we're happy to have you here, and we hope you find this session to be helpful. Speaking of helpful, a lot of our coaches and students have used USMLE-Rx to their benefit and to succeed on exams. So we'd like to know, do you have a USMLE-Rx subscription? Do you have one? Do you not have one? Are you in a trial? If you don't have one, you may be one of the lucky winners tonight that gets wins one of our raffles. We've got some raffles coming up at the end, and so stick around. You might be the lucky winner, and sure enough, every week we have a winner or so that isn't here. So we have to draw another name. So don't be that person. All right, I'll give you guys a couple more seconds to respond. And then we will move on and begin our question dissection on genetics. All right, let's take a look here. So it looks like over half of you guys do have it. Excellent. And it looks like about 41% of you guys don't, and 4% of you guys are in the trial. So for you 41% that don't have access, you should join the 4% at least and take advantage of our free trial. It'll give you access to all the product lines, RX 360 Plus, which includes RX Bricks, our question bank QMax, our Express videos, and our flash facts. So I encourage you to sign up for at least a free trial to experience it like so many other students have, and perhaps you'll be able to use it on your path to 250. I briefly mentioned RX Coach. RX Coach is brought to you by the authors of First Aid and USMLE-Rx. It is our one-on-one coaching program, and we work with students individually to identify their strengths and weaknesses and work on their test-taking skills and timing and stamina. We've had a lot of success with it, and we have great coaches just like Ari, who you met briefly a moment ago, and we'll talk more about RX Coach later. And the methodology that we're going to use in tonight's question lab as we dissect these 4 genetics questions will be the same methodology that we use in RX Coach. And so with that, I will hand it off to Mr. Jeffrey Downing.
Okay, it's time for question number 1. So as we open this up, you're going to see something that might strike you. Where are the answer choices? Well, this is a part of our process. We don't include the answer choices to start. We want you to focus on the question first. We cover up the answer choices so that we don't guide or dictate your thinking going into the question. We also don't want you to see a word or an answer choice that you're completely unfamiliar with, or you've completely forgot. We don't, because that might cause panic on test day. So that's what we do. So we start by looking at the lead-in. So in this question, the lead-in question is, during which of the following steps of cell division did the child's defect most likely occur? So now we're going to read the vignette. A male infant is born at 40 weeks gestation by normal spontaneous vaginal delivery to a healthy 38-year-old mother. On examination, the neonate has a flat facial profile and prominent skin folds at the inner corners of the eyes. The infant fails to pass meconium. A loud S1 with a wide fixed split S2 is appreciated on auscultation at the left upper sternal border. So again, our question then is, during which of the following steps of cell division did the child's defect most likely occur? And now we can look at the answer choices.
Excellent. And before we do that, I want you guys to think about, is this a 1-step question, a 2-step question, or a 3-step question, right? So an example of a 1-step question could be where they ask you for the diagnosis. An example of a 2-step question could be where they ask you, what is the treatment for a diagnosis? And an example of a 3-step question could be, what is the mechanism of action for a treatment for a diagnosis? So with that in mind, shoot us a response in the question box and let us know, how many steps do you think this question will require? Is it a 1-step question, a 2-step question, or a 3-step question? And we'll wait for your responses, and then we'll go over that and uncover the answer choices. Excellent. So we have a lot of people saying 1-step, 2-step, or 3-step. This is probably a 2-step question, right? First, we got to figure out what the diagnosis is, and then we got to figure out, when did this defect occur? So with that in mind, an organized thought process, which is very important for question dissections, let's go ahead and take a look at the from E to A. That's what we always like to do. And that forces you to go through all of the answer choices, make sure you don't skip any, but it also prevents you from rushing to judgment and selecting something without reading all the answer choices. So choice E, telophase 1, D, prophase 1, C, metaphase 2, B, metaphase 1, and A, anaphase 1. So I'm going to pop open a poll here and give you guys a couple of seconds to respond and select track what they think the diagnosis is, but that's not what this question is asking you. It's asking you, when did this defect occur? So we'll give you guys a couple of more seconds to respond. Remember, on test day, you should never leave a question right. So make sure you never leave a question blank on test day. And right now, if you're thinking about this and you're just not sure, you haven't looked at this material in so long, don't worry, take your best guess. We're going to go over the correct and incorrect. Just we'll give you 4 seconds. Then we will go over the correct and incorrect answer choices. All right, let's see how you guys did. Looks like 35% of you guys picked metaphase 2, 27% of you guys picked metaphase 1, and 24% of you guys picked anaphase 1. So let's see what the correct answer is. The correct answer is A, anaphase 1. Okay, so it looks like 24% of you guys got that right. So congratulations to all of you. If you guys didn't get it right, don't worry, pay attention, and you'll be able to get it right on test day. But most importantly, master it so you can become the physician you've always dreamed of becoming. So this patient has Down syndrome. And we know that because the diagnosis was pretty straightforward, right? They told us in the question that he had some of the classic features and characteristics of Down syndrome, including a flat facial profile and prominent skin folds at the inner corners of the eyes. And they told us that he fails to pass meconium. They even told us that he had a loud S1 with a wide fixed S2 at the left upper sternal border. So what are they talking about? They're talking about epicanthal folds here. That's what these are. That's what these folds are. And when they say that he didn't pass meconium, they're hoping that we're going to be able to deduce that this patient has Hirschsprung's disease. And this, of course, here, the loud S1 with the wide fixed split S2 at the left upper sternal border, that is an ASD, or atrial septal defect. Now, those aren't the only side effects and symptoms that you see, the symptoms that you see with Down syndrome. There are other ones like intellectual disability, stunted growth, hypotonia, duodenal atresia, as well as brush field spots. And those are those white gray spots that are on the periphery of your iris, as well as a single polymer crease, okay? Now, why is it anaphase 1? Well, it's anaphase 1 because in approximately 95% of cases, Down syndrome is due to non-disjunction during anaphase 1 or 2 of meiosis, okay? Now, during anaphase 1 and 2, a microtubule spindle separates chromosomes to opposite poles of the dividing cell. Down syndrome results when the copies of chromosome 21 fail to segregate normally during anaphase, eventually leading to a zygote with 3 copies of chromosome 21, aka trisomy 21 in Down syndrome. Now, there is another genetic cause of Down syndrome. Can you guys tell me in the question box what that genetic cause is? We'll give you guys a couple of seconds to respond. I'll give you a clue. It starts with the letter R. Excellent, and we have some people that took the bait. It's Robertsonian translocation. That's correct. So, you know, we can also have Down syndrome in people who have Robertsonian translocations, but that is pretty rare, okay? Now, having Down syndrome, that risk will increase as the mother's age increases, okay? And that's because a majority of infants with Down syndrome are born to younger mothers, okay? So do not let the fact that she is under 40 years old throw you off, okay? So the correct answer here is anaphase 1. And notice that anaphase 2 wasn't listed, so that is probably the most likely and correct answer. Now, let's go ahead and go over the incorrect answers. Telophase 1, so that is the stage in meiosis 1 during which the first physical division of the cells occurs. And that's when each daughter cell is containing one homologous pair of chromosomes. And that's not what's happening here. Prophase 1, that's the initial stage of meiosis 1 and the stage during which the replicated homologous pairs join to form tetrads, okay? Metaphase 2 of meiosis 2 is the step during which the sister chromatids line up along the central metaphase plate of the dividing cell. And metaphase 1 is the step during which the homologous pairs of chromosomes line up against and along the central metaphase plate of the dividing cell, okay? So the moral of the story here is that 95% of the cases, Down syndrome is due to non-disjunction during anaphase 1 or 2 of meiosis. And here is a little slide that kind of go over Down syndrome. As you can see, they told us he had flat facies, epicanthal folds. They even told us and gave us clues about Hirschsprung disease and ASD, okay? So they had a lot of clues here that pointed us in the direction of Down syndrome, and it was our job to figure out what caused it. In 95% of cases is due to meiotic non-disjunction, and in 4% of cases is due to unbalanced translocation, aka a Robertsonian translocation. So wonderful job to those of you who got it right. For those of you who didn't, hopefully you guys learned from that question and will get it right when you see it in the future. Jeff.
Okay, that was a great start. Good challenging question to get us kicked off. So let's go into question number 2. So again, we've got the answer choices covered up, and we're going to start with the lead-in. Based on the observations, which of the following regions of the lac operon most likely contains a loss of function mutation? Now, the vignette. While working in a microbiology laboratory, a researcher tests whether a strain of E. coli has an intact lactose operon. After growing the cells in media containing both glucose and lactose, she observes that a significant amount of galactosidase encoded by the lac operon is expressed compared with controls. Galactosidase production is also observed in the presence of lactose only. No protein products are produced when the E. coli is grown only with glucose. So again, based on the observations, which of the following regions of the lac operon most likely contains a loss of function mutation? Sean?
Excellent. Thank you, Jeff. So this is a complicated question. So let's think about how many steps this question requires. And normally in our coaching sessions, what we'll do is we'll stop after every sentence and kind of pick your brain and ask you, hey, what's important here? What are they telling you? What's on your list of differentials? What could be causing this? Because we don't have time to do that today. We'll do our best to explain this to you when we explain the answer choices. But for now, let us know, how many steps do you think this question will require? I'll give you guys a second to respond, and I'll uncover the answer choices. Looks like we have some people saying it's a one-step question or a two-step question. Yeah, it could probably be a two-step question, right? So we need to figure out what's going on, and then we need to figure out what caused it and what's going on here and causing this loss of function mutation. So either a one-step or a two-step question. So great job to all of you. And with that thought process in mind, let's take a look at the answer choices. Once again, we'll start at answer choice E and work our way up. E, repressor protein. D, RNA polymerase. C, promoter region. B, inducer binding site. And A, catabolite activator protein binding site, or CAP binding site. So I'll give you guys a second to take a look at our poll and select what you believe is the correct answer. And then as always, we will go over the correct and incorrect answer choices. Now keep in mind that when you're doing questions on your own, it's very important to review why you got it right and make sure you got it right for the right reason, and also understand the incorrect answer choices and what in the question made the right answer right and the wrong answer wrong, so you can work on those test taking skills. And once again, make sure you stick around until the very end. Jeff is going to draw some names, and we're going to have a wonderful raffle for you guys. Looks like the responses are a little slow to come in. It's probably because you haven't reviewed this material in a while, which is why we're doing it today. So we'll give you guys a few more seconds to respond, and then we will go over the correct and incorrect answer choices. All right, looks like the answers are coming in. Let's see what you guys selected and how you guys did. So 28% of you guys picked A, which was catabolite activator protein binding site. 31%, in first place was 31% repressor protein, and in third place was the promoter region. So let's take a look and see what the answer choice is. And the correct answer is A, and 28% of you guys got that right. So good job with the catabolite activator protein binding site. So here is a diagram here of the lac operon. What is a lac operon? The lac operon is an inducible set of genes that allows bacteria to use lactose as energy when glucose is not available. So when glucose isn't around, the lac operon allows bacteria to use lactose as energy instead. But when glucose is present, the lac operon should not be active, and that's because cyclic AMP levels will be low in the presence of glucose. And when cyclic AMP is low, it does not bind to CAP, and thus transcription of those genes involved in lactose metabolism is not activated, okay? So let's take a look here. So the lac operon is a gene segment, like I said, that encodes for important parts of lactose metabolism, including those for galactosidase, okay? And galactosidase, if you guys remember, will cleave lactose into glucose and galactose, okay? So when glucose is absent and lactose is available, the lac operon is activated to switch on this metabolism, okay? Excellent. So in this vignette, it's important to notice some of these clues. they tell us that when it was grown in both galactose and lactose, there was a significant amount of galactosidose encoded by the lac operon, right? And they said that it was also observed, galactosidase production was also observed in the presence of lactose only, but when it was grown only with glucose, no protein products were produced, okay? So those are kind of your key things to keep in mind. Now, in this vignette, galactosidase is not produced when E. coli is grown only with glucose, since lactose is not available to bind to the repressor. When E. coli is grown in the presence of lactose only, galactosidase is produced. But in the presence of lactose and glucose, galactosidase is still produced at a high level. And this suggests that glucose is failing to suppress the expression of the lac operon. Therefore, the mutation is most likely located in the cap binding site, okay? Now, if you're looking for this in first aid, it's going to be found in your genetics section, and look up E. coli. You'll be able to find this diagram, or go to the index and look up lac operon. It's always a subject that gives students a little bit of a hard time, but we're going to go through these incorrect answer choices, and that'll give us a review of the lac operon. So the repressor protein, what does it do? Well, it functions to bind the operator site on the lac operon and inhibits protein production. Now, lactose will normally bind to the repressor to prevent this binding to the lac operon. So if the repressor were mutated and non-functional, we would observe protein production in any condition where there is low ATP availability, regardless of whether we have lactose or not. Now, the repressor in this vignette is functioning normally because protein production is inhibited in the glucose-only situation and permitted in the lactose-only situation. So that's how we can rule out that the answer choice E is incorrect. Answer choice D, RNA polymerase. Well, RNA polymerase is responsible for the process of transcription, which is generating RNA by using DNA as a template. If RNA polymerase was mutated, all gene expression would be affected. And we did not see that either in this vignette. Answer choice C. Now the promoter region, okay, the promoter region is located upstream of the lac operon, where RNA polymerase will normally bind. So if the promoter were mutated, lac operon transcription would vary based on the mutation. For example, a severe mutation could prevent RNA polymerase from binding altogether, while a less severe mutation could potentially decrease or increase the affinity with which RNA polymerase binds to that promoter. A mutation in the promoter region of the lac operon is not what we're seeing here, so that is the incorrect answer as well. Answer choice B. The inducer region, what is that? The inducer binding site is where lactose, the inducer of the lac operon, will bind. If the inducer binding site were mutated, the lac operon would not be expressed in the presence of lactose because the repressor protein would remain bound to the operator region, okay? So that's a mini review of the lac operon, okay? And answer choice A we talked about was the correct answer. All right. So great job to those of you who guys got it right. I see a lot of people saying the lac operon confused them when they were studying, and that's, you know, me and you both. Okay, so it confused all of us. But there's a really good description of it in first aid. So take a look at that, and let's move on now to the third question of the evening. But before that, I'm going to turn it over to Mr. Jeffrey Downing.
Well, thanks, Sean. And, you know, we just wanted to take a minute and remind you about RX Coach. This is our one-on-one tutoring service. A lot of the strategies that we talk about each week are derived from, you know, what we do when we're working with students. And, you know, so we've been doing this for over a year and a half now, working with students specifically on step 1, but also we've had students come in looking for assistance with step 2 CK, with the the CBSSAs, or just trying to work through some of the issues that students typically find challenging in med school. So we have a process in which when you get started, we have a 160 question assessment that allows us to see what your strengths and weaknesses are, helps us develop a personalized study plan with you. And then, with our RX certified tutors, and these are, med students just like you, a little bit older. They've been through the process and have done really well on their step exams. They're the ones that will help kind of lead you through this. And really, one of the differentiators is that we have RX 360 Plus, which includes the bricks and includes COMLEX. So we're able to pull from an authoritative tool to help ensure that you're on track. So you're going to be able to get access with that. Now, some of you, might be thinking you don't need a coach, or that coaches are only for the students who are really struggling, but that's not really the way to think about coaching. World-class performers depend on coaches to help them improve and maintain their high standards, right? So Serena Williams has a coach, Roger Federer has a coach, every golfer on the PGA Tour has a coach. Coaches are there to make sure that you are performing at the top of your game, and to help you through those stretches that are struggling. So if you're going to be successful as a physician, you really need to be seeking out mentors at every step of the way. And RX Coach is designed to provide that level of mentorship. So, if you find yourself thinking that you could, really benefit from some one-on-one coaching, give us a shout. If you head over to rx-coach.com, you can set up a free consult. And if you're ready to up your game, we're going to pair you with an experienced coach like Ari, and then set up a plan for success. So in the chat box, we have a link to RX Coach. So take a look, and if you if you're ready, we're ready to work with you. So check it out, Sean.
Awesome, thank you, Jeff. We're going to move on to question 3. So I will let Jeff kick it off.
Okay. Well, everybody, Gregor Mendel would be very, very pleased with the work you guys have done tonight. Let's see how we do with this question. So I'm going to start again with the lead in. Which of the following represents the likelihood that this couple's child will be affected by cystic fibrosis? Okay, so here's the vignette. An Ashkenazi Jewish couple expecting their first child came to the physician. Both parents are healthy and have no medical conditions. The mother's family history is remarkable for a brother with cystic fibrosis. Her parents are unaffected. The father's brother also has cystic fibrosis. Upon his brother's diagnosis, the father was tested and found to be a carrier of the Phe508 or PHE508 gene mutation. The mother has not been tested for genetic mutations. So again, the question is, which of the following represents the likelihood that this couple's child will be affected by cystic fibrosis? Sean.
Thank you, Jeff. So in this question, we get to do every medical student's favorite thing, right? Which is math. We're gonna have to do some math to get this one right. But don't worry, it's not too complicated. Before we get to the answer choices, instead of me asking you this time how many steps you think this question will require, I want to ask you how cystic fibrosis is normally inherited. So I'll give you guys a second to enter your answer in the question box and let us know what the inheritance pattern for cystic fibrosis is. Because that's the first step. We got to figure that out, and then we got to do some math afterwards. So this is likely a 2-step question. So we'll give you guys a couple of seconds to submit your responses here. Excellent. And I'll even throw you guys a bone here, since I know you guys don't like math. And I see a lot of people saying autosomal recessive, and that is correct. It is inherited with an autosomal recessive pattern. And with that in mind, and that little clue to help you out, let's read these answer choices. Answer choice E, 50%. Answer choice D, 3%. Answer choice B, 17%. And answer choice A, 100%. So this question is gonna probably take you guys a little bit of more time to figure out because you guys have to do some math. So we're gonna give you guys a second here, or a little bit more time than normal, to do the math here and figure out what you think the correct answer is. And while you guys do that, I will tell you that we have had a lot of success with RX Coach. So if you guys are interested in that free consultation, like Jeff said, go to rx-coach.com to discuss how the program can benefit you along your journey like it has so many people. We've had so many students score above that 250 mark, and they never thought it was possible. We had a student that wanted a 230, came to us, and she got a 250 plus, and she said, well, maybe I can break a 260, and she in fact did, okay? So to discuss our program and how it can benefit you, visit us at rx-coach.com. If you want some free generalized study advice, we have a lot of that too available at firstaidteam.com. Give you guys a couple more seconds to respond here. I see you guys are crunching those numbers and selecting those answer choices. And I already gave you one clue, which was you have to do some math, which automatically should remove some of these answer choices from selection. And I'll go over why that is the case here in a moment. All right, let's take a look and see how you guys did. Looks like 59% of you guys chose answer choice C, which is 25%, and 27% of you guys picked answer choice B, which is 17%. Excellent. The correct answer is B, 17%. So let's see how we got to that answer. So this is an autosomal recessive inherited disease, okay? And as you guys know, cystic fibrosis has symptoms like secretions in the lungs, the pancreas, the liver, the intestine, the reproductive tract. And we normally recommend newborn screening if any parents have a positive family history for cystic fibrosis. Now, in this case, they told us that the child's father, okay, they told us that the child's father had a history of cystic fibrosis, and they even told us that he is a carrier of cystic fibrosis, going so far as to even confirm that he is heterozygous for the PHE508 gene. And we see that right here, okay? Now, we know he's heterozygous because he didn't express the disease. So that means that he has one mutant allele and one normal allele, right? Because he's heterozygous, so he gets one normal allele and one mutant allele, all right? So the probability of passing on that mutant allele is 1/2 or 50%, right? Because he has two alleles and his chance of passing on that one allele for cystic fibrosis is going to be 1/2. Now, although the mother did not have testing to determine her genotype, they gave us the phenotypes of her family members, and we can use that to get a better picture. So we know that she is disease-free. So because she is disease-free, she cannot be homozygous for the mutant allele, right? Everybody on the same page? Now, if she was homozygous, she would have cystic fibrosis, but we know that her brother, however, is affected, but that her parents are not. So even though she has not been tested, okay, we know that the mother's family is remarkable for a brother with cystic fibrosis, and her parents are unaffected, okay? Now, that means that because the brother has it and she does not, we can conclude that she is probably a potential carrier. Now, her unknown genotype could be any of the following three, right? She could be heterozygous with her mother's mutant allele. She could be heterozygous with her father's mutant allele, or she could be homozygous for the normal allele. Let me repeat that. We have three possibilities of her genotype here. She could be heterozygous with the mother's mutant allele, heterozygous with her father's mutant allele, or homozygous for the normal allele. Now, she is not a carrier in only one of these three possibilities, which is that she carries a single mutant allele in the other possibility. So if you look at this, in 2 out of those 3 situations, she is a carrier. So in 2 out of those 3 genotypically possible situations, she is a carrier. So what's 2 over 3? That's 66%. So we're going to take that 2 over 3 and put it here, okay? So now we have to figure out the combined probability of the mother being a carrier and then passing it on. Well, we know that the probability of her being a carrier out of those 3 situations is 2 out of 3, which we've written down. And her passing that allele then on is also 1/2. So we multiply that by 1/2, and we're left with 33%. Everybody follow? So now, in order for the child to have cystic fibrosis, which is an autosomal recessive disease, okay, they must have a mutant allele from each parent. So since the father, we already said, has a 50% chance of passing on that allele, okay, and the mother has a 33% chance, we multiply 33 by 1/2 and get the answer of 17%, okay? Now, hopefully you guys followed the math there. So it's 2/3 from the mother with 50% chance she could pass it on, and 50% from the father. 33% multiplied by 50%, or 1/2, is 17. Now let's discuss why the other answer choices are incorrect, okay? Let's discuss why the other answer choices are incorrect. E, 50%. Well, 50% would be the risk to the child affected when one parent is affected with an autosomal dominant disorder like Huntington's disease. And that's not what we're dealing with here, and it's not the mode of inheritance for cystic fibrosis, okay? 3%. 3% is the approximate recurrence risk for the offspring with an individual that has a multifactorial genetic disorder like cleft lip or cleft palate, right? And this can also apply to the siblings of the affected child, all right? But cystic fibrosis does not exhibit multifactorial inheritance. And I also told you guys that we have to do some math, so that should have ruled this answer choice out for you guys as well. 25%, we already went over E and D, let's go over C. 25%, that is the risk of the child being affected with an autosomal recessive disease if and only if both of the parents are confirmed to be carriers of the diseased allele. And that is not the case here. We did not confirm that the mother is in fact a carrier. We said she might be a carrier, and that might probability was 2/3, okay? So that's why answer choice C is incorrect. B, we went over, was the correct answer, 17%, and we showed you how we got to that number. A, 100%. Well, this is talking probably about mitochondrial inheritance. And once again, doesn't include any math, so that trick should have helped you out. But in 100% affected rate, okay, that's mitochondrial inheritance, and that is because mitochondrial inheritance comes from the mother. So 100% of an affected mother's children will receive that disease, and none of an affected father's children will receive it, because children will inherit their mother's mitochondrial DNA, it's maternal inheritance. And cystic fibrosis does not exhibit mitochondrial inheritance, which is why answer choice A is incorrect, okay? Excellent. Let's move on now to the last question of the evening. Before we do that, if you guys like how we're dissecting these questions, showing you the clues and breaking it down for you, if you think you could benefit from coaching, visit us at rx-coach.com. What we like to do is start with an assessment, gauge your strengths and weaknesses, and then make you a study plan so that we can guide you, make your time efficient, make your study prep efficient, and maximize your potential. And as we start to pick your brain in these question dissections, we start to know what kind of learner you are, how you think, and what your test-taking deficiencies may be, so we can finesse those test-taking skills and help you on your path to 250. So you can sign up for the free consult to discuss that program at rx-coach.com. And with that, I'll hand it back off to Jeff for our last question of the evening.
Okay, Sean, let's close this one out strong. I know we've had some good, challenging questions tonight. And as Sean always says, it's, hey, it doesn't matter if you get it wrong tonight. It's the making sure that you get it right on test day. So here's our final question of the night. We're going to start off with the question itself, the lead in. Which of the following best accounts for this observed pattern of inheritance? Classic genetics question here. So here's the vignette. A 64-year-old woman comes to her general practitioner for a yearly physical examination. She says that 20 years ago, she was found to have a BRCA1 gene mutation. However, She is in perfect health otherwise. 3 of her 4 children were also found to have the same BRCA1 mutation. One of them was diagnosed with prostate cancer, while another was diagnosed with breast cancer. The 3rd child with the mutation is in good health, and the 4th child is not a carrier of BRCA1 or a mutant gene. The pedigree is shown. Which of the following best accounts for this observed pattern of inheritance? Sean.
Excellent. Thank you very much. So we'll go into the answer choices now. Once again, we always start with answer choice E and work our way up. And the reason for that is because we want you to look at every single answer choice, not rush to judgment. And this will force you to go through all of them to make sure that you're not getting tripped up by your nerves or taking any shortcuts on test day. So answer choice E, variable expressivity. D, loss of heterozygosity. C, incomplete penetrance. B, dominant negative. And A, anticipation. So we'll give you guys a moment to select your answer choice. And then we'll go over the correct and incorrect answers. We'll give you a couple minutes, a couple seconds to respond here. I see the answer choices coming in. Once again, we have some challenging questions today, but that's okay because we can learn from them together and hopefully get it right on test day. I see a lot of questions about the math that we did earlier, and if we have time at the end, I'll touch base on that for you guys briefly, okay? So don't sweat. All right, let's take a look and see how you guys did. 48% of you guys picked answer choice C, incomplete penetrance, and 32% of you guys picked variable expressivity. Let's see what the correct answer is. And the correct answer is C, incomplete penetrance, and 48% of you guys got it right. Our best performance of the evening. So congratulations to all of you guys for getting it right. For those of you who did it, like I always say, we're going to learn from the explanations and hopefully get it right on test day. So let's talk about why this is the correct answer. So a 64-year-old woman we know has the mutation, okay, but she is in perfect health and doesn't have any associated health problems. Then they tell us that 3 of her 4 children also have that same mutation. One of them was diagnosed with prostate cancer, while another was diagnosed with breast cancer. The 3rd child is in good health, and the 4th child is not a carrier of the gene. So in a presentation like this, we must first consider the pattern of inheritance. From the pedigree alone, it could be mistakenly assumed that the BRCA1 gene mutation is autosomal recessive. However, they tell us in the vignette that 3 of her 4 children carry the mutation. And out of those 3 affected children, only 2 are symptomatic. So it's safe to assume that the BRCA1 mutation is inherited in an autosomal dominant pattern and exhibits a phenotypically variable presentation. And we call that incomplete penetrance. So in essence, a mutation may be harbored and inherited by a family member, but the expression of it depends on the extent of its penetrance. And the risk of expressing that phenotype then becomes the percent of penetrance multiplied by the probability of inheritance, okay? So this is why the correct answer is C, and 48% of you guys got it right, so well done. Now, why are the other answer choices incorrect? Well, variable expressivity, what is that? That's a condition in which patients carrying the same mutant genotype have varying phenotypes, right? So they will all have the same genotype and they'll have varying phenotypes. So an example of that could be tuberous sclerosis and neurofibromatosis, in which the individuals share some or all or no common traits. And that's not the case here. This is unlikely as 2 of the affected individuals are in perfect health and fail to express any trait specific to this BRCA1 mutation, okay? So that's why it's not variable expressivity. D, loss of heterozygosity. Now, this is when an individual who previously lost an allele, and that's usually they lose it congenitally, and the last and final allele is due to like a additional insult, something else happens. So this occurrence is what we call the two-hit hypothesis, in which two alleles is required to activate a proto-oncogene, like in retinoblastoma, okay? And that's a well-known example because in retinoblastoma, these mutations can either be sporadic or inherited. And the inherited form will result in a heterozygous allele configuration at the time of conception. And then you have another additional insult, which is required after birth to lose that heterozygosity and permit that unregulated cell cycle to continue. So loss of heterozygosity is unlikely in this pedigree because not all the individuals carrying the mutation are affected by the mutation, okay? So this is the two-hit hypothesis, and that's not what we are seeing here. C, we discussed that, and that is in fact the correct answer. So great job for those of you who guys got it right. B, dominant negative. What is dominant negative? Dominant negative is a term used to describe a heterozygous allele that produces a non-functional protein. So when that protein is non-functional, it prevents the normal gene from functioning properly. And a common example of this is familial hypercholesterolemia, which leads to hyper LDL because those LDL reuptake receptors are not properly functioning. So this is describing a non-functional protein, and that does not account for this because some individuals exhibit no effects at all from the gene mutation. So we can rule out dominant negative. Once again, dominant negative is used to describe a heterozygous allele that produces a non-functional protein, and that is not the case here. Anticipation. What is anticipation? Answer choice A. This is usually used to describe conditions that are inherited autosomal dominant as well, but that increase in severity and present with earlier onset in subsequent generations. Can you guys give me an example of a condition that displays anticipation? I'll give you guys a couple of seconds to respond in the question box and let us know. Excellent, I see it. Fragile X syndrome, Huntington's disease. Great job, guys. And Huntington's is one of those most popular examples of it. So what happens then is in Huntington's disease, a trinucleotide repeat expansion will lengthen with each generation of offspring. So while this condition is widely affecting a family with an autosomal dominant inheritance pattern, not all of the individuals carrying the mutation are affected, and we have no reason to believe that the presentation is occurring earlier with a more aggressive progression, which is what would happen with anticipation. So with anticipation, it gets worse and more severe and tends to happen with an earlier onset, and that is not what we saw in this vignette. Now, I see a lot of you guys asking about the 17%. We got that 17% in the previous question from 2/3, which was the mother's chance of being a carrier, multiplying that by half, which is passing on that allele, okay, which is then 33%, and then 50%, which is the father's chance of passing on that one allele. So 50% multiplied by 33% is how we got to that 17%. All right, and with that, I will show you guys new resource that we came out with not too long ago, but it's something that's been helping a lot of students. And I want to make sure you guys are familiar with it because you guys can actually try it out for free online. Maybe not this brick, but we have several bricks that are available for you to access. So this is an RX brick on cystic fibrosis. And these are these learning modules that are very interactive and help you master the material. So they start off with learning objectives, and you see 4 of them defined here for you. And then you can practice and learn with this module, what is cystic fibrosis? And then they ask you these questions where you can flip and really start to flip these cards and test your knowledge as you're reading. We have some nice diagrams there. And then we even have, towards the end here, clinical correlations, another clinical correlation, how to treat it, how to diagnose it, and then we have a summary of the take-home points that you should learn from each brick. And once you learn from the brick, you can practice with these practice questions. And once you get the right answer, you will get the explanation. And we even have this humanism perspective, which gives you some real world examples and helps you relate this content. Then you can review these learning objectives and mark off which ones you're comfortable with. And of course, like today, if you have any questions on what we talked about, you want to go deeper, you can click on the brick and it'll take you directly into first aid. You can click on flashbacks to review it quickly. And flashbacks has that study stream where you can see these your weaker topics more often due to spatial repetition. We also have express videos, which help you master this content and really give you some amazing explanations of the content that you see in first aid and in bricks. And of course, once you feel like, hey, I've got this, let me go practice it, then you can go to COMLEX and practice that knowledge and make sure that you have, in fact, mastered that material. So if you haven't signed up for a free trial already, feel free to do that at USMLE-Rx.com. As always, we want to encourage you guys to stay connected with us, get social with us. If you guys are interested in RX Coach, you want to have that one-on-one coach to maximize your potential, visit us online at rx-coach.com. Sign up for a free consultation to discuss the program and how it can benefit you. If you want some generalized study advice, we have a lot of that available too. We even have a free study planner you can download at firstaidteam.com. If you guys like this webinar, if you guys like the content we put out, including like Mnemonic Mondays, practice questions on Instagram sometimes, follow us on social media. We're on Facebook, we're on YouTube, we're on Instagram, okay? So follow us there. We're even on Twitter. So stay connected and you'll learn about some cool upcoming events like the webinar we have tomorrow at 8 p.m. with our learning specialist to help talk to you about learning strategies that you can use in medical school. As always, we do these webinars, these question labs, Tuesdays at 6 p.m. Eastern. And next week, we'll be talking about neuropathology. So we hope that you guys will be able to join us then.